folks, and welcome to the Emeriti Association membership meeting and presentation for the month. I'd like to just mention that this is the season, a season that we haven't really celebrated before. It's the season of getting immunized against coronavirus. And hopefully all of the members of the Emeriti Association and any visitors from the Retirement Association have thought that it was the best thing to do, which I believe it has been, and are getting their vaccines if they're in the correct age group. I know there's lots of stories about where to go and what to do, and UCSD has been in the forefront of immunizing folks. According to the town hall meeting that I attended last week, UCSD has actually given more vaccines than all the other UC facilities in the state. And it's a real credit to the administration and the folks in UCSD Health that this has been an accomplishment. And it's a partnership with both the state of California as well as the county of San Diego. So a number of members of the Emeriti Association and the Retirement Association have in fact volunteered to help out at one of these sites. And if you have thought about it but have not already done so, then please make the effort to look into it and to sign up so that you can help out in this monumental effort. I'd like to pass the gavel, so to speak, to Stephen Adler, the Vice President of the Emeriti Association this year, who's also the chair of the program committee. And he's going to introduce our speaker for the month. Stephen?
Thank you, Jake. Good to see everybody here, and I know our numbers will grow over the next few minutes. And so I will vamp for just a couple of seconds and say that we're more than halfway through our program for the year with our distinguished speakers. We have Professor Trey Ideker today, and I'll introduce Professor Ideker in just a minute, but I'd like to remind you that next month in March, we have Professor Kim Cooper from Division of Biological Sciences talking about how and why the jerboa got its long feet. In April, we will hear from Professor Amelia Glazer in the Department of Literature talking about Yiddish songs and poems from World War II, and I believe from other times as well. And then in May, guest visiting Professor Harry Littman will be talking about restoring the rule of order in a post-Trump world for our big May end of year lunchtime gala. So I would urge all of you to mark those. You'll be getting notifications, of course, from Suzanne. I see there is a question from Margaret. Was that in for this conversation that Margaret Huff? Yes, Margaret, you're muted. Margaret, you're muted.
I didn't have a question. I was just pushing buttons.
Oh, okay.
It's only the second time I've done Zoom.
That's okay. Welcome aboard.
Okay.
Great. Well, then without any further comments or questions about the rest of the program, I would like to take the opportunity to welcome and introduce my friend, Professor Trey Ideker. Hello, Trey. It's great to see you, even if it's under these circumstances. And I will just give you a little bit of Professor Ideker's background biographically. He is a professor in the departments of medicine, bioengineering, and computer science here at UCSD, and director or co-director of 3 NIH-supported research centers, the NIGM's NIGMS, National Resource for Network Biology, the NCI Cancer Cell Map Initiative, and the NIMH Psychiatric Cell Map Initiative. He received his bachelor's and master's degrees from MIT in electrical engineering and computer science, and his PhD from the University of Washington in molecular biology under the supervision of Dr. Leroy Hood. Dr. Ideker is a pioneer in using genome scale measurements to construct network models of cellular processes and disease, and has founded software tools, including the Cytoscape ecosystem for biological network analysis, which has been cited over 13,000 times. Dr. Ideker serves on the editorial boards for Cell, Cell Reports, Molecular Systems Biology, and PLOS Computational Biology, and is a fellow of AAAS and AIMBE. He was named a top 10 innovator by Technology Review and was the recipient of the Overton Prize from the International Society for Computational Biology. His work has been featured in news outlets such as The Scientist, San Diego Union Tribune, Forbes magazine, NPR, and the New York Times. So please join me in welcoming Professor Trey Ideker. The Zoom floor is yours.
Well, thanks very much, Stephen. It's a pleasure to be here, and I wish I could be there in person and give a live dynamic lecture, but here we are, and we have what we have, don't we? We will still have a good time. So I'm going to try to share my screen and then go into present mode. So let us try this. Okay, can someone give me an indication that you can see? All right, I'm getting, give me thumbs up. Excellent. Yes. So definitely stop me. You know, sometimes you find that when speakers, they march along and they think that the audience can see what they see. And in fact, the audience does not see advancing slides. These things do hang. So if you get the eerie feeling that I'm talking about something that you aren't seeing, please do stop me and let me know as I go along. And Stephen, just to get the parameters, so the floor is mine for how long?
I would say for about 35-ish minutes, if you'd like, and then we'll take some questions and answers afterwards.
Yeah, so, and I can adjust as we go on, depending. I mean, I'm also happy to take questions during, as the case may be, and you know, I'm happy to adjust, and we can do time checks as we go. So as the title of my talk suggests, I'm going to talk today about the intersection of two important topics. One is cancer, and what are some of the both advances and continued challenges in understanding and treating cancer, and two is the intersection of understanding cancer with the advances in artificial intelligence, and that's going to prompt me to give some background information about what are those advances in artificial intelligence that have gone on over the past couple of years that have, you know, given rise to any numbers of news releases that even if you're not in the field, you must have heard about. Things like, you know, beating of Go champions and chess champions and self-driving cars and things like that. So let me, and actually, if you're in a biological field, the most recent advance of artificial intelligence is what's called the protein folding problem. It was recently announced by Google, for instance, that they had essentially solved a 40-year longstanding challenge in biochemistry, which is the ability to take any sequence of a protein and determine its three-dimensional structure through their deep learning systems. And you know, as time goes on, we'll see how accurate that claim is, but it looks like it's so far pretty accurate. All right, so let's get started. First of all, before I talk about how AI is impacting cancer, let us just make sure we're on the same page with fundamentals of how people think cancer occurs and what is cancer as a disease and as a mechanism of disease-causing agents. So although people still argue about this and probably always will, the current party line is that cancer is a disease of mutations to your DNA, the so-called genome. The textbook picture that would describe that is shown here on the slide, as long as my advancing of slides is working. So you have a normal cell population, and this, by the way, this describes generally cancer as it forms in many, if not all, different tissues. Of course, there's different variations depending on whether we're talking liver cancer or blood cancer or so on and so forth. But in all of those cases, we have a normal cell population that over time accrues mutations. These are often called somatic mutations as opposed to germline variations in your DNA, because in most cases, not in all, but in most cases, we're thinking about mutations that happen throughout life. These aren't things I was born with. You know, I get melanoma maybe because, you know, my arm here is getting UV rays on it all the time. Now, many times I see that I get moles, and those do not go on to form tumors. But certainly there have been mutations that have impacted my skin cells, which are then causing or underlying some of those moles, even much less melanoma. Worried about getting here in sunny San Diego. But so mutations are raining down on our cells and our bodies all the time. It is in a sense the unfortunate event that some of those mutations embed in genes in a way that they cause the cells to multiply in a way that's abnormal. A normal way that cells are supposed to multiply is wound healing. I scratch my arm, and of course I should be able to regenerate that tissue. In a sense, what's happening is regeneration has gone awry, and now you're going to get a tumor mass where none was expected, certainly, or welcome. And so that's what's shown here on the slide. And as that process progresses, there's research into how many such mutations, unlucky events you need, and lots of evidence that you need more than one unlucky event. So it's sort of a chain of unfortunate events that occur to give you this malignancy. And then ultimately, we talk about a localized tumor, even if it's malignant, then can escape and become metastatic, meaning that now it will appear in distal sites. So what was a breast tumor is now become a bone tumor, and vice versa. And that's, of course, in a sense, the worst of all possible worlds in development of cancer. So there's my Cancer 101. And if that wasn't clear, please do shout out. Now, what has been the advance of the last, say, 20 or 30 years in terms of cancer, our understanding of cancer and where exactly the mutations occur? If I was to do this talk in person, I would make this a Socratic type of method where I asked as many questions as I spoke. But maybe because we're in a Zoom format, I'll just tell you some of these answers. And so who knows when the first human genome was sequenced? Let me put it that way. Before we talk about cancer genomes, you know, anyone who's not a biologist, I guess, when did we have kind of the big announcement that we could sequence human genomes? Maybe we'll, you know, I'll see if any brave soul wants to unmute, or Stephen, or do you have control over that anyways? This is not going to work.
No, I don't, but somebody just posted something in the chat.
Oh, my chat is totally not visible to me. Hold on. Yes. Richard, I believe, got it correct, that it was 2001. And in fact, there were two genomes. It was there were two enormous efforts. One was a public project by a company called Celera. The other was funded by the National Institutes of Health, so the federal project. And essentially, there were two papers back to back that essentially had two different versions of that first human genome. And it was later revealed that Craig Venter, who was behind the company, it was actually his genome that was published. So since 2001, let's now talk about how that genome technology. So that people have estimated that the 2001 human genome, which was the culmination of 20 years of research and development by thousands of people, if not tens of thousands of people. The price tag on that was certainly well in the millions, if not hundreds of millions, or maybe I've even seen numbers as high as billions of dollars for that first human genome to just get humanity to the point of sequencing people's DNA. But now let's talk about what happened after that time. And what happened after that time is over the next 10 years, between 2001 and I'll say 2010 or 11, we saw, just like people talk about Moore's law and microchip technology and how it becomes better, faster, cheaper to make computers with some doubling time of the number of transistors and a halving of the cost and all of that. The same kind of rule of Moore's law has absolutely been used to describe what occurred with DNA sequencing. And so by the mid 2000s, by 2005, 6, this technology came along that utterly revolutionized the speed and lowered the cost with which DNA can be sequenced. And it was called the sequencing by synthesis method, and the company was Illumina. So we're now like 2006, 7. Illumina, as you may, well, who knows where Illumina is located? Let me put it that way. And you're free to give as an exact answer as you would like. Hint. Yes, it is. Joan is correct. Now, Joan, where exactly is it? I would say UTC. Basically, if you drive to the Crate and Barrel next to UTC Mall, you take a right if you're coming from the university, and you're immediately on the Illumina campus, and it just gets bigger and bigger and bigger. I think now that if you're on the 805 going north, you see this huge Illumina sign and whatever. And so all of that growth that's happened in just literally right off of our campus was the pivotal moment was this release of this new sequencer device by Illumina in the mid 2000s. So now all of a sudden, people, and of course there's other advances all the time. I'm belittling as I tell the story, but I think it's somewhat faithful. And now all of a sudden by the late 2000s, you have the ability to sequence people's DNA for a few $1,000s, and of course the cost continues to come down. So we've gone from hundreds of millions of dollars for the first couple of genomes to now a couple of $1,000s for your very own genome. This is when things get interesting with regard to applying this weapon to understand disease. Cancer being one of the most common diseases and one of the largest killers in not just the US, but many countries, has been one of the early foci of what can we do with sequencing people's DNA? Well, since cancer is supposedly caused by mutations in DNA, it would seem that a natural match would be to sequence a bunch of cancer patients' DNA and look at what was found there. So that's exactly what happened. And so by now, late 2000s and early 2010s, this project had started called the Cancer Genome Atlas, or TCGA. If you go to these conferences, people speak in acronyms, and one of the acronyms you'll hear a lot is TCGA. Major initiative of the National Institutes of Health. At this point in time, this project is largely wrapped up. It sort of completed its mission, but certainly by 2015, say, and by the present time, I like to think about maybe the TCGA started in the late 2000s, wrapped up just a few years ago. So that's another decade or so that's passed. We now have 10,000s of examples of cancer genomes. It is not yet to the point where your insurance would pay for that cancer genome. It's in a gray area where if the doctor orders it, they'll sequence a few hundred genes that are thought to be particularly important in cancer. What anyone in the field will tell you is, well, gosh, what we really should be doing is just spend $1,000 and sequence the whole kit, caboodle of your genome. Don't just sequence a couple things you think might be important. That's not yet paid for insurance. What is a couple hundred genes, but nonetheless, for research purposes, government dollars and company dollars have paid for a few tens of thousands of complete tumor genomes, and that's what people like me study. Okay, so there's a gap between clinical practice still and what we actually study. That nonetheless, if you now look at those data, and by the way, is this one cancer? No, this is when I say 10,000 genomes, I'm talking about 20 different cancer types that have been surveyed in this way. So you can do the math. It's hundreds of examples per. Breast cancer probably has the most right now. It's probably over 1,000 individuals' tumors that are in that database. So that's a lot of data. And so this plot that you're looking at as I'm speaking is an attempt to summarize the main findings. It's always good to kind of go from storytelling into actual data. And so this is what you're looking at. Oops. I did not mean, whoa, hold on. There we go, I'm back. What you're looking at here are the genes, some sort of infamous cancer genes and genes that are in the data are mutated a lot. Those are these symbols like TP53, VHL, RUNX1, these are all, or RB1, these are all well-known cancer genes, many of them. And then the other axis here is the cryptic code that is used in these projects for the tissue type. So OV is ovarian. I mentioned breast cancer is BRCA, and so on. Bladder is bladder at BLCA, and so on. And then the height of the bar is the percent of patients of that kind of cancer for which a given gene is found to be mutated. So if you look at this, what's by far the scariest gene, or at least the most frequently mutated gene in any cancer type practically, and that is P53 over here at the far left. You can see in almost all, certainly and let's look at it at ovarian cancer. Okay, if I look, that's his back corner. P53 is mutated in nearly all of the cases that have been examined. We always see in ovarian cancer P53 gene mutated, but you see, there's a lot of cancers that have to mutate P53 as a way forward for the tumor to grow. And it's not just P53, and so this just gives you some insight into some of the top genes that have been found. Many of these were known already, many of them weren't, and so those would be the novel discoveries out of these kinds of projects. Okay. Feel free, as always, feel free to stop me if you need to. So now I mentioned that there was a gap between sequencing all of your DNA and sequencing a few of these cancer genes only. And so here is what the clinical version of this looks like. There's companies. This is not the only one, but you can see I'm on the FDA website here. So this is an FDA approved diagnostic for cancer mutations, and that has come out of some of these sequencing projects like I just described. This one happens to be called the CDX diagnostic, and it's by this company called Foundation, and the product is Foundation 1. Okay, so how is this now supposed to work? Well, the way it's supposed to work with genomics is that you now sequence everyone's DNA, or everyone in your cancer ward, and for each patient, this one's the example is a breast tumor, you would discover what genes are gone awry, have these mutations, and that would then and by the way, then down this lower pathway, this patient actually has become metastatic. So what was originally a breast tumor is now spread to the brain. Definitely not good. But what you would do is you would take the gene mutations that you find, make designer drugs, or just select already available drugs against those mutations. And you would first test in a mouse that drug hypothesis is going to work. So you would basically take a sample, you'd biopsy the tumor, or you'd remove the tumor from surgery if it's possible, and at least the primary site. Now, one of the problems with metastatic cancer is it pops up all over the place, so surgery seldom works at that stage. But certainly at the primary stage, you would remove the tumor. You can certainly remove a metastatic site as well, although it's not going to cure the cancer. And you'd implant those in mice as a sort of proving ground for your drug hypotheses, and then you would then try over the since you can grow and perform these experiments in a matter of weeks to months. It's often the patient, you would have time to do that and actually see what works in your mouse and what doesn't work, and if it works in the mouse model of your disease, because this is after all your tumor embedded like under the skin of a mouse, then you would then be able to try that drug in the patient. That is how all of this, that's the vision of where all of this is supposed to go with cancer. This is what essentially is called precision medicine in the cancer, as applied to cancer, when you hear things like precision medicine used. Okay. So why isn't it all just, why don't we have this right now? Why isn't this all working, as planned? There are a number of reasons we could talk about. One of the reason I'm going to talk about today, and one of the most often talked about reasons, is what is known as heterogeneity. So what is heterogeneity in terms of cancer mutations? What it means very simply is that if you look from patient to patient, as is shown over here at the left, even those would say now here we're looking at different, in this pictorial graphic, you see the patients have different tumor sites. But even if you were to pick like breast cancer, where everyone supposedly has the same tumor type, as you now look at what genes are mutated, you actually find we saw before kind of a summary across many cancer types what genes tend to be mutated, but from patient to patient now, it can be very, very different. And in fact, many patients will have mutations that have never been seen before. That is, so we mentioned how in ovarian cancer, you're almost guaranteed to have P53. But now, what I didn't tell you then that I'll tell you now is there's another 100 genes that are mutated besides P53, many of which are seldom seen, if ever seen, in other cancer patients. Said a different way, all of our, we know that we're all individuals, all of our, and in genetics, we reflect that by saying, we all have different DNA sequences. They all have common sequences that makes us human, but then of course we're all different individuals of the human species, and we look different and we act different. And so our DNA can't exactly be the same. And it's that rule that we're all snowflakes genetically applies to cancers, unfortunately. Every cancer is an exquisite snowflake, never seen before on Earth. And that therein lies the problem. You may find a couple of genes that you recognize that are hit by mutations, but you then have to deal with this sea. And if you're unlucky, you don't even find those. And then you always have to deal with this sea of so-called rare mutations that have never been observed or rarely been observed. And what the slide here is showing is that these differences actually even exist in a single tumor. You can't even think about a single tumor as I'm going to drug that mutation, because in fact, as you look around the tumor, you often find that the tumor is really a bunch of little tumors, each one of which is its own little genetic snowflake. And so maybe designing a drug against the tumor is just going to take out this tiny bit. And not to mention that these tumors are dynamic landscapes as well. And so what mutations exist today may be a different landscape as the tumor evolves to become resistant to the drug you just gave. So it's this all of this maps back to the problem of heterogeneity. These are not the same genes it's not the same formula that's giving everyone their cancer when it comes to DNA, at least. It's different. There's going to be many different pathways to cancer. So that suggests, does that indeed suggest that maybe we need a separate drug for every person, and a separate complete study, essentially a little scientific study you have to do every time to figure out what in the world is causing this person's cancer, and that's going to be unique for that person. That would be the extreme. People are hopeful, and I'm hopeful that there are common pathways to cancer, but certainly we're going to have to grapple with this issue of so-called heterogeneity. Right. So that's I think I've now spoken everything that this slide was meant to express. There were just too many patterns, or too many maybe already abandoning hope, but certainly there are a great many patterns of cancer forming mutations, and we only understand at present a few of these patterns. And so how we're going to grapple with this extremely complex landscape of ways of causing cancer is the challenge. Here's a sort of, in the field at least, famous quote by Eric Lander, who I mentioned. Craig Venter was sort of a leader of a company that sequenced the first human genome. Eric Lander was one of the leaders, and still is, of the public project to sequence the human genome. You might have actually read Eric Lander is now our science advisor, and he's on President Biden's cabinet. So that just shows, what sequencing, the first human genome can do for your career and your impact on science. But the quote, which is the important point, lest I get ahead of myself here, is human genome, bought the book, difficult to read. Okay, so now this would be another good time if you have any burning questions to ask, because now this is where artificial intelligence comes in. Why does it come in? Go back. I just said there's a lot of complicated patterns. Artificial intelligence, in a sense, is exactly good at finding signal in complicated patterns. That's exactly what artificial intelligence does well in certain other domains, and we'll talk about some of those in just a second. So let's now switch gears and have, like, it's almost a separate talk in a way, and then at the end we'll merge these two directions. So what do I mean specifically when I'm going to talk about artificial intelligence? Of course, I think I just outlined the question of today's talk. The central question of this whole 35 minutes or so is, can modern AI be taught to read the cancer genome and eventually tell you how you got your cancer and what drug to give? That's the idea. Now I can see the time. I am at least 20 minutes into my talk here. So we'll see how far I get towards solving this solving cancer with AI. But let's proceed regardless. The idea would be you'd have some machine here, and I have this black box here in the middle, and I feed it your mutations of say you're a cancer patient. I give it your mutations, and it's going to give you your drug and what kind of cancer, what manner of cancer you possibly have. Okay, so why is there optimism that artificial intelligence might be helpful? Well, certainly we've all read and heard about all of the recent impacts of so-called machine learning on other areas of life. game playing, Jeopardy, I think was a number of years. Chess and Jeopardy were now a number of years ago. Some of you may remember all the news around those. The most recent game playing success was now still a few years ago, where the so by the way, in order of complexity, Go is considered more complex of a game than chess. And so it took a little longer, supposedly, for machines to beat people. But then just a few years ago, the Go champion of the world was beat by a machine. Certainly, the fact you can now talk to Siri is aided and abetted by speech recognition, and if you compare it to Siri's ability to understand, it's still, as you may notice, Siri is still not great sometimes, but at understanding you and transliterating your speech, but she is certainly way better than what existed 10 years ago. Image recognition is another huge area where artificial intelligence has driven a lot of excitement. More on that in just a second. Certainly self-driving vehicles, especially with what Tesla has done to make us appreciate self-driving, even if we're not quite there yet, I think has nonetheless been really exciting. Now, those are all outside of medicine, but even within medicine, there's been a lot of excitement mainly in biomedical imaging. You might have seen work where, speaking of cancer, you can show you can upload a picture of your not melanoma, but of your skin, of any lesions you're worried about, and you can have a machine that the idea is in the very near future you will have a machine that can give you an opinion about whether that's a problem, and then you can go see your doctor. That actually works, which is pretty exciting, powered by some of the advances in deep learning. So here's an example where imaging, machine learning and imaging affects our daily lives without question. Anytime you Google an image, so you hit the images tab as opposed to the all search, right? And so if I type cat into Google Images, I am continually amazed at how accurately it returns cats. It is no longer, once upon a time in the recent past, it was just reading like the captions, and it was literally just a text match. But now it's absolutely the ability to, and you've heard about facial recognition and essentially the social, the scary social aspects, the Orwellian, one might even say, aspects of that. Maybe a benign, same technology, more benign case is this one. Let's just search for cats. Look how darn good this thing is at finding cats, and I've just shown you the top dozen or more. But imagine if you just keep scrolling down, you would find this thing is 100% accurate for thousands and thousands of results. So why can't we simply sort of do that metaphorical thing for your cancer genome? Just learn how to say, here's the genome. Just tell me where are the cancers in those genomes, and where are the responders to this therapy, or where are the people who really are at risk, and where are the people who are probably should not be so concerned, and so on and so forth. One of the problems, I'm going to go through a bunch of problems, but one of the big problems here is there are groups. We're working on that problem. There's a bunch of groups working on that problem, and the machines are getting pretty good at telling whether or not a cancer is going to respond to this particular drug regimen or this other particular drug regimen. The problem we now have is these are all researchy types of systems. They haven't yet been moved into the clinic, and part of it is people are mistrustful of the machines, as well they should be. These are all so-called black boxes. You can't ask the machine, why is that a cat? Why is this a cancer patient who is going to respond to palbociclib versus not respond to palbociclib? It's incredibly hard you can't simply open up the box and interrogate this thing. It doesn't know. It just gives you an answer, and that's that. You have to accept it. So just accepting a machine's answer is really, really difficult, especially when you have high stakes applications like you're treating patients with drugs. Getting, mistaking a cat for a lion, that's a pretty low stakes mistake. Self-driving is a little higher. Cancer therapy is the absolute highest stakes possible. So we're going to have to start getting some insight into what these machines are actually doing and thinking, rather than just accepting their answer. Here's a classic case where the answer is, it can give you the right answer, but if you actually, when scientists, in this case, artificial intelligence researchers, tried to understand what was actually going on, they realized it was incredibly misleading. So here's a case back from image recognition where it wasn't recognizing, the problem wasn't cats, it was dogs, and distinguishing between dogs and wolves. Google Images and the machine that these researchers used, very, very good at distinguishing dogs and wolves, okay? And here's some examples. So, starting from the left, here's a wolf, there's a husky. You can kind of see why it might be difficult, but the machine gets it right. Then moving over, that's a wolf, and so on. Well, let's move down. See where the X is? It gets the X wrong. Okay, that is actually a husky, okay, not a wolf. And when the researchers went and tried to finally figure out what rule is it using, if I was to ask you, for instance, how do you know that this is a wolf versus a husky? You would presumably say something like, there's something about the head shape or the ears or the eyes or the look of the thing. You're focusing your attention in a reasonable way on the body morphology and the face especially. How did the machine actually do it in the end of the day? It looks in the lower left corner for whether it sees white, indicating snow. And if it sees snow, it says wolf. And if it sees green, it says dog. And so anyway, the machine was right, but it was right. It got it wrong finally when it was surprised. But it fooled people for a long time into thinking it was very, very accurate, when in fact it was, but for entirely the wrong reasons. So this is sort of the pitfall that we really want to avoid when you just unleash these types of systems on human disease. So Stephen, I'm going to try to finish up here in just a slide or two, because I know that we're getting kind of, or at least come up for air. We're kind of 30 minutes in or so. I think what I'll do is I'll just skip to kind of just to give you a flavor of what we're doing, maybe is the best use of the time here. And so essentially let me maybe back up. What we're doing is we're trying to force these machines to have rational, so not just predict what tumors based on their DNA, what tumors are going to respond to a drug or not, but to have some knowledge of basic cell and tissue mechanisms, basic biology that guides their reasoning. And it's this guiding of the reasoning that's going to be critical for avoiding crazy rationale that, it's akin to looking at the lower corner and seeing white and declaring that to be a wolf. That's not what you want here. You want to be able to tell patients and their physicians that, hey, you had a mutation in Notch or in TP53 or in these other famous cancer gene. And we have a drug that can attack that mutation, and that's how your treatment is going to go. You need to have that sort of knowledge of the reasoning of the artificial mind, not just the answer. So to do that, we've been baking into our artificial minds, our AI systems, knowledge from basic cell biology. We go into cell biology textbooks that have diagrams like this one of how genes connect to other genes. We also try to flesh out these wiring diagrams through primary experiments of how genes connect to other genes. And this knowledge is exactly what gets used inside of our AI systems. So, and you know, I think the best way is just to illustrate how this works. So in other words, rather than just feeding some black box up here at the top, the cancer patient genome and a potential therapy you're trying to give, and expecting it to give some answer, which is a yes or no. Yes, this patient is going to respond, or no, this patient is not going to respond. That would be the output. We're trying to put a rational logic and knowledge of the way the cell works and the way the tissue works into the middle of the system. And that's what these funnels are meant to describe. I don't expect, given my level of explanation here, you to understand or me to be able to describe exactly what goes on at the next lower level, but you should sort of just get the notion that what's happening is we're injecting knowledge into the system. The fact these funnels are nested inside of one another represents the fact that machines like cells and tissues are very similar to man-made machines in that they're exquisitely hierarchical. So you have genes encode small parts, which are then parts of bigger parts, which are then parts of still bigger parts, and cell, which then become assembled to become cells, and cells assembled to become tissues. And so you have this exquisite hierarchy of small to large machines, and that's all part of this knowledge base that you're giving this machine. And this notion of how you might, it turns out this notion of how you might do this kind of learning is actually very old. It's my favorite review article in this field is by this guy, Herbert Simon. He was a Nobel laureate in economics, in fact, but he's almost equally known for his work in computer science and artificial intelligence. He was a professor for a long, long period of time at Carnegie Mellon University in the Department of Computer Science. This one particular piece called The Architecture of Complexity, where he ranges from man-made systems like watches. I think his first example is a watchmaker, to biological systems like cancer cells, and then back again to man-made systems. He does a beautiful job of painting the commonalities of all of those complex systems. And central to his entire piece here is, and of course, I think he has economic systems also in this review, but central to this entire, whether it's Wall Street or watches or cancer cells, is this notion of this hierarchy of machines that beget bigger and bigger machines. And that's absolutely stimulated our work in how we think about guiding these AI systems. And so here is just, I'll end with just kind of one example. Oh, sorry, I see I have a question. How do we know from Jacoby, how do we know if there is a role for epigenetic factors giving input to the genes rather than the genes themselves? So that's a great question. I have completely, for those of you who know something about epigenetics, which is controlling, so I guess the easiest way to explain it for those of you who haven't heard this term is, in addition to whether you have mutations in the genes themselves, you also have the issue of how much is that gene expressed? Is it making a lot of protein, or is it, so genes make proteins which do things in cells. So is that gene turned on to a high degree, or is it turned off? And that it's epigenetics that really concerns the degree to which genes are active or inactive and how that process happens. That's another huge area of cancer research, and epigenetics, absolutely, the degree to which a gene is on or off, irrespective of the genome, whether it's been mutated or not, is a huge area in cancer research. The reason I'm not addressing it here is with gene expression, there's been less of an issue of so-called heterogeneity. You don't have to grapple with all these rare events like you do for mutations. With gene expression patterns, it turns out you can make sense of them a bit better. You can see clear clusters of this set of patients expresses these genes, or the second set of patients expresses these other genes. So really, it's the mutations that are both supposedly closer to the root of cancer and also more cryptic from that point of view. But certainly, everything I'm talking about, one would like in the future to then move into the epigenetic layers. Okay, so here's just one example of kind of what, just to give you kind of a flair for what the readouts of these systems become. And so now here is an example where this tumor got paclitaxel. Okay, where that was we had a whole panel of tumors and both in cell lines and in tumor models that had been exposed to this drug paclitaxel. Taxol is more colloquially known simply as Taxol, Taxol. It is a FDA-approved drug. It's a very reasonable line of therapy for several cancer types, including breast cancer, I believe also ovarian cancer, among others. It had been thought to work by binding the structure of cells known as microtubules and making them very rigid. And so somehow this is killing the cancer cell. But people argue about it. And there's been a number of papers that took issue with how paclitaxel actually worked. More important, a lot of patients don't respond to paclitaxel. So the more we can figure out about what it's actually doing or failing to do in someone's tumor, the better. So here, our system that we designed was not only able to correctly predict whether a group of patients was going to respond to paclitaxel, given here not the epigenetics yet, but the mutation pattern in their DNA. Given that, we got a prediction that turned out to be pretty accurate. And that then led us to look at why. So now we have not just the will a patient respond, but we begin to get at the why. And the why, it turned out, was as much involved in another process related to what's basic metabolism, glycolysis, as it was to the supposed mechanism related to microtubules. And so here is just sort of a part of the wiring diagram that's in the inner guts of our model. That's kind of, and you can see innervating that diagram here are the internal circuits that our AI is using to make its predictions, which are correct. And you can see they involve names like response to glucose, insulin secretion, involved in response to glucose. So and response to cyclic AMP is another glycolytic related mechanism. So what it appears to be is that mutations in glucose sensing and metabolic pathways are what may be beyond or behind whether your patient is going to respond to this drug. And then we've gone on to test that in lab, and that's what's shown down here in the bottom right-hand corner. We can muck with glucose metabolism of your cancer cell, and with there's this sort of fake or a mimetic for glucose called 2-deoxyglucose or 2-DG. And what we can show is that if you apply in the first column here, you apply just the paclitaxel drug. In the second, you apply the glucose mimetic. But when you combine them in the third column, you can see a whopping killing effect on cells. This is not a patient, by the way, this is cell cultures. Nonetheless, it's encouraging. And we've gone on to kind of repeat this exercise a number of times with different drugs. And the processes in the cell, it looks like, are important for modulating whether or not you're going to get a response. So that's that. I think I'll basically wrap it up. Just in summary, really, if you had to kind of summarize my talk in just 2 sentences, it would be something like, separately the fields of genomics and the field of artificial intelligence. I think we're all aware how exciting and how rapid the advances in both of those fields have been. It is certainly the case that they have not been integrated to their maximal extent, and I think we're even kind of in the early days of seeing what happens when you apply AI to analyze disease, patterns in disease like mutations. And so I think we're just at the dawn of enormous progress and certainly experiencing presently lots of opportunities in how this goes down. If you want to find out more about what we're doing in particular, you can just, I think like all of us, at UCSD, there's an obligatory web presence of some sort. You can find me easily enough. I didn't at all talk about the one time in my career I've been in People magazine. Was we had this completely unrelated, speaking of epigenetics, by the way, we had a completely unrelated result where we used epigenetics to show that dogs do not age as we thought they did with that 7-to-1 rule, 7 human years per 1 dog year. It turns out is not really correct. If we look at the molecular level, it turns out dogs age way more rapidly than Tom Hanks, but people. It's a non-linear aging relationship. Anyway, I won't belabor this point, but if you're interested in a cute, and a bunch of cute dog photos, an interesting read, you can check out this story as well. It certainly is. This is a story that bizarrely spun out of our cancer research 8 years ago, and this is what it's become. So please check it out. And I probably have a picture of my lab. Okay, well, there we are, Stephen. We're hopefully not crazy. We haven't tried people's patience too much, and we can take any further questions, or or take it wherever you like. I'll turn it back to you.
That's great. Thank you so much, Trey. That's terrific. Really, really marvelous presentation. So I would like to turn it over to Suzanne, because she's the best moderator for the Q&A process.
All right,
process
but
here.
beware, I do not speak epigenetics or AI, so the best would be for people to raise their hand electronically. We'll recognize you and you can present your question yourself. I know we have a number of doctors online with us, and I don't want to have to interpret your question for you.
Well, and of course here, I'm sure I'm not the first or last speaker to realize. You're caught between people on this call who know more than you do about this subject, and people on this call who know very little, who know much less. And so let's see what happens.
All right. So Rick Bolin, go ahead.
He says no microphone, but he's chatting his question, and it says, how do you actually solve a problem using AI? I'd have to ask him what he means by that. So in a sense, so Go, the game of Go is solved by beating your opponent. And here, in this case, the game of cancer, of your cancer genome or your disease, is solved by coming up with the correct drug to administer. But is that actually what you're asking?
Can you hear me now?
Yes. Yes.
Okay. I just plugged in. I was just listening passively. So for the problem of genomics and a drug, do you just put in one end, I don't understand what it looks like inside the black box. So you put a lot of DNA sequences in from the tumor and then put outcomes in and just do it over and over and over again.
Oh, okay. Well, actually, so I think you're asking about the training. So
Yeah.
Hold on. I would argue that for this, I of course could give more details, but this slide was the closest to what the inside of that black box looks like. And so above here, you have what's not shown here, I guess, is right up here above. You have things like mutations that are, raining down on these different processes like glucose response. Sorry, there we go. Clicking and pointing, it depends on where you are on this thing. So above this, you have mutations hitting these different processes, and then the outcome is going to be at the bottom. Somehow the AI is going to synthesize all of this into an answer, which is going to be paclitaxel is going to cause a response in your tumor to regress, or it's not. You're going to be non-responsive. But when you asked your question, you said, how do I train? What I heard is, how do I train this model? And the answer is, we start in domains where we have a lot of data, which in drug screening is cell lines. So what drug companies and basic scientists like myself and other labs are very good at doing is taking cancer cells, in a dish, so to speak, and seeing what happens when you expose those cells to thousands of chemical compounds. And so essentially, and then you can do that for thousands of cells, each of them having a different genome, each of which has a different set of mutations, a mutated gene. So the way I like to think about it in terms of like math is, you have like a matrix of thousands of tumor genomes that in this case are in cell lines. They're cultured as cells, and each of those 1,000 tumor samples, quote unquote, or tumor cell samples, is exposed to say 1,000 drugs. So if I do the math, 1,000 by 1,000 is about 1 million data points, where I have the whole input would be the entire set of mutated genes, and the output is for that, well, and the drug, that's also the input. And then the output is did the cell grow fast or slow, essentially. It's actually a quantitative number, how fast the cell grew. What's called the area under dose response curve. So you have like order of a million training examples from these cell culture experiments, or high throughput drug screening, as it's called in the pharmaceutical industry. And as you may know, these AIs, they suck up data, so they're very good at, reading millions and millions and millions and millions of Twitter feeds and deciding how to give the most caustic possible robotic response that's going to cause the most people to do what they want, right? I mean, that's exactly what's happening on Twitter right now, and they feed off of millions and millions and millions and millions and millions, right, of Twitter conversations. Here, you're feeding off of millions of mutation patterns and drug responses. Now we move into, I mentioned briefly at the beginning of my talk, you can move into mice as kind of an intermediate between a patient and a drug administration. And so an intermediate playing field is what are called PDXs, which are patient-derived xenografts in mice. And there you have a moderate amount of data, where you have, here is the genome of the patient's tumor that was put in a mouse, and here's the 30 compounds. So then they give they create 30 mouse clones, all of which have this patient's tumor, or they just simply, I think often you'll just see that they take 30 little bits and put it in different parts or different mice, and then they give them 30 different drugs, and so that's like a moderate amount of data for which you have drug responses for a few hundred or maybe a few thousand, but not millions of example tumors. And then ultimately you want to move into the clinic, where you have the least data still.
Trey, may I ask, please? This is actually medicine that's being practiced, that's being utilized now.
No. So everything I'm talking about is still preclinical, although we are starting. We're starting clinical trials with. It looks like, fingers crossed, with pharma and also internally at Morris Cancer Center, but they're just getting off the ground. And of course, the challenge right now you are in this what's called the gap between basic research and actual clinical success. That's this sort of valley of death that's been much written about in the pharmaceutical industry. And so we're attempting to navigate that valley. We're trying to fund a clinical trial with Barbara Parker at the Cancer Center as we speak. What you can do, and by the way, everything I'm talking about, I should say, was I kind of flashed a publication at some point on a screen. I think it might have been back a slide. How do I do that? Yeah. everything I'm talking about here is this, see this first underlined thing, Kunze Park et al. Cancer Cell 2020. This all just came, it's literally hot off the press. It just came out in November as the base. So you know, definitely preclinical, but we're trying to move it into the clinic.
But
Yeah.
you were saying that basically it's all going to be private pay. So this is designer treatments available for the wealthy.
Well, that gets in. That gets into something much larger than this particular talk and my research, right? That's how do we get a world where insurance pays? Well, how do we get a world where you get you get medical care? Everyone gets medical care. That's even a larger order question. But the sub question is, how do we achieve a world where you can get your cancer sequenced and get your insurance to pay for it? That's a battle that I think can be won in the next couple of years. That's kind of the where I think we really need to push. I think the way to win is for people like me and my clinical counterparts to provide ever more overwhelming evidence that knowledge of your DNA absolutely affects your outcomes. And certainly it's happening all the time. So for instance, if you have a mutation in breast cancer, if you have a mutation to BRCA1 or BRCA2, BRCA1, BRCA2, these are genes that I'm imagining a lot of people have heard of. They're infamous breast cancer genes. That's determined by sequencing the BRCA gene. If that happens, then you qualify for a drug known as olaparib, which targets another part of the circuit that typically compensates for the BRCA1 mutation, and so you kind of give it a lethal combo that it can't tolerate. That's a perfect example of an FDA-approved drug that's now a major type of cancer therapy that is made possible by the cancer genome and that knowledge. And there's many, as time goes on, there's many of these examples emerging.
Thanks for addressing that. Any other questions from our members, please? Everyone's in awe.
Well, it's also getting near, it's 4:52.
So nobody else?
I have a question actually, and it's tangentially related to the presentation, Trey. But what spurred your passion for this particular avenue of research?
Great question. That's a question that can be answered on so many levels, Stephen. I'll answer it on a very early, at least time point. So you mentioned kind of something about my background when you introduced me, and so what I forget was in there, if it was in there, is how I started in terms of subject matter. And so I did not start in biology. I started in computer science. My undergraduate degree was in computer science. I ended up as a newly minted undergraduate working. So back then, this was like 1995, 4. Back then, this was pre.com boom. If you can believe it, the internet, I think, was right around 1994, or not the internet, the World Wide Web. The internet was this DARPA funded thing that, you sent email and went into, FTP sites and chat rooms and things, but there was no web. The web had just hit, but no explosion yet. So computer scientists went one of two ways. You went to Wall Street or you went to defense. I went, speaking in broad strokes, I went to defense, and I had been there a couple of years. I was working for a contractor off the 128 Beltway in the Boston area at the time. I was disillusioned. I enjoyed sort of the day-to-day, but I literally had a philosophical problem that I couldn't imagine, ending my career having built bombs for a living, and because that's essentially what I was doing, and I just couldn't handle it, and so I had kind of a career meltdown and crisis at the age of 24, and that induced me to apply to then sort of take some time off and do some soul searching. I think I did some traveling probably that year, and ended up applying to grad school in biology to try to kind of do the opposite thing. And part of that decision was I had a friend who was one of my best friends from university was an immunologist at that time, and he said, Trey, people like you might end up being quite valuable as this thing called the Human Genome Project gets underway. And so I graduated, so I got my PhD in that same, speaking of when the first Human Genome Project or first human genome was published in 2001, as was correctly answered, that was when my PhD was awarded. And so I kind of started, and then I started here as a professor in 2003. And so if you think about my career, it's really been kind of, timed with the rise of genomics and driven by the rise of genomics since that 24-year-old career crisis.
Well, thanks so much, Trey, for sharing that and the rest of the presentation. You should know that it will be viewed fairly widely once it's posted to the Emeriti and Retirement Association's YouTube channel. Have a very big viewership there. Just please, everyone, join me in thanking Professor Ideker for his time and his passion and his wisdom on this. And Jake had one last quick question. What cancer do you think, in 30 seconds, what cancer do you think will benefit most from the AI procedure?
I think a number. I think the answer is a number here. We're out to really, this is bigger than one cancer, but I think especially those tissue types like brain tumors, where biopsies are very difficult, and I think also stand the most to benefit, provided you can actually sequence their DNA. But that's another conversation. It turns out there are ways you can sort of access that. So basically, cases where where surgery really is not very successful, so you really have to rely on this, what's normally the second fallback, which is chemotherapy.
Thank you. And Rick makes a point, maybe by sorting tumors according to mutational signatures. I should let you folks have at it at another time and place amongst yourselves. I think it's great to have these questions come in, but we're at the end of our time. So I will again thank Professor Ideker, and feel free to drop him a note of thanks. We will see you all, I hope, in March for our Professor Kim Cooper. Again, Trey, thank you. It's great to see you, and thank you very much, everybody.
Thanks very much.
Thank you. Thank you.
Bye-bye.